to Matthew Harris's presentation. So Matthew, as a student in the biology department, who has been working with me over the last year and a half maybe, looking at interactions within the soil of dwarf mistletoe. So please help me welcome Matthew Harris.
Thank you. Last talk of the day. Thank you all for being here. So I'm sure many of you are like me, since we live in this wonderfully awesome state of Colorado. You probably enjoy being outdoors, hiking, photography, bird watching is one of my favorites. But if you spend any time to the west of us in the San Juans, you probably notice the immense amount of trees that are dead and dying due to the spruce beetle infestations. Another parasite that may be less obvious to us is the dwarf mistletoe. My name is Matthew Harris, and I want to share with you the effects of dwarf mistletoe on the soil ecosystem and how that affects the soil microbes that live in the soil and how they cycle the nutrients. Just an outline of where I want to go in this talk. I'm going to introduce the key organisms in this system and tell you how they all interact with each other and how they help or not help each other. Then I'll state our purpose, followed by our methods of this experiment, and then our results, and then we'll have time for questions afterwards. So the first key organism in this system is Pinus edulis, or pinion pine, also known as the Colorado pinion pine or the two-needle pinion pine. Interestingly, fun fact, this is actually the state tree of New Mexico. So we can fight over pinion pine and whose chili is better, I guess. This tree is a short to a medium-sized tree. It's a long-lived species, living up to 800 years. A fairly drought-resistant species also. You'll notice this pinion jay has this pretty big seed in its mouth. This species of tree is very important ecologically to the animals in their environment. These seeds hold about 3,000 calories per pound, and on a good year, the trees can give off about 300 pounds per acre. So a very great source of nutrition in this ecosystem. This tree is also the host to our second key organism in this relationship, the dwarf mistletoe. Dwarf mistletoes can be dispersed by the birds that eat their seeds, or the very sticky seeds can stick to the fur of rodents or the feathers of birds. But the main mode of dispersal is by hydrostatic pressure, where water pressure builds up behind the seed and it shoots off like a cannon onto neighboring trees. They do this in the fall in the rainy season, and then when the sticky seeds stick to the needles of the neighboring tree, they slide down the needle and embed themselves on the branch. And then over winter, they just kind of hang out there, and in the spring, they'll germinate. They have a root-like structure that penetrates the tree and starts stealing nutrients and water and carbon as well, because dwarf mistletoe is not very efficient at photosynthesis, so it also relies on the tree for carbon. After germination, about 2 years later, you'll start seeing swelling. That's the first evidence of infection. After that, 2 more years, the shoots start to appear. And then it takes 2 more years for the parasite to be sexually mature and restart the cycle. Dwarf mistletoes can infect various conifers, and each conifer has 1 or 2 species that are known to infect it. Our Colorado pinion pine is infected by Arcithobium divaricatum, and here are a bunch of infection sites that are known to exist. Here in the southwest Colorado and Four Corners area, they're infecting our Colorado pinion pine, and further west they infect the single needle pinion pine, a close cousin. Because this parasite is stealing nutrients from the tree is going to have higher needs for nutrient uptake from the soil. It also has other effects. Defoliation is a major effect, reduced or distorted growth, and eventually in some cases death. But these effects vary from tree species to tree species and the species of dwarf mistletoe and so on. So not all trees are affected the same way. Our Colorado pinion pine is not heavily defoliated and rarely dies from infection, but dwarf mistletoe infections can hasten or worsen the effects of other antagonists, such as the bark beetle or drought, and that is our real concern, especially with the pinion pines, because we are in a drought here in the Southwest. The effects that we're interested in are the nutrient cycling in the soils, and because soil microbes are responsible for the uptake and transport of those nutrients to the tree, we should see effects in the community structure of the soil fungi that live in the soils associated with those infected trees. Pinion pines, other pines, and conifers in general have an ectomycorrhizal relationship. It's a symbiotic relationship with fungi, where the fungi will infect the roots of the tree, effectively increasing the surface area of the roots and increasing nutrient uptake, transport, and water uptake as well. Because the dwarf mistletoe is stealing nutrients from the tree, there's eventually going to be reduced nutrients in the soil that can lead to competition here in the soil fungi, and that should change the community structure to species that are more capable of efficiently taking up those nutrients and delivering them to the tree. But also, because the parasite dwarf mistletoe takes carbon from the tree, the fungus also takes carbon from the tree. So it's going to be getting less carbon off the roots. So there's going to be competition for carbon there at the roots, which can also affect the community structure out here in the soil, which we're going to investigate. In 2015, Dr. Duran and Ryan Schilling did a study of dwarf mistletoe infections on soil concentration, nutrient concentrations in soils associated with ponderosa pine. And what they found that infected soils had increased nitrates, increased phosphorus, and slightly decreased potassium concentrations in their soils. So because of these effects, the fungus is responsible for moving these, so we should see, like I said, a change in their community structure. The purpose of our experiment is to find those changes, except this time we're going to look at pinion pine. And remember that dwarf mistletoe is taking carbon as well as nutrients from the tree and water. And then the fungus also takes carbon, but it's giving water and nutrients. So this is a very complex relationship that takes a lot of thinking about sometimes to keep your players straight. So the methods of our soil sampling, you may have noticed in that infection map that the San Luis Valley doesn't have any infections close by. So we went to New Mexico, just north of Taos in the Carson National Forest. And there we sampled 12 infected and 12 uninfected trees on a 12-foot transect. Every 2 foot, we took a sample of soil and pulled it together for our nutrient concentration analysis. And then at the 4-foot mark, we took an extra soil sample to do a molecular DNA analysis to identify the species that are living there in that soil. So after we brought those soils back and cleaned them up, we sent them off to get analyzed for their nutrient concentrations. And what we got back were concentrations of phosphorus, potassium, and nitrates. For our methods on the molecular analysis, we took that soil sample and we extracted the DNA from it. And then we did a polymerase chain reaction to increase the concentration of this specific region of sequence in the fungal genome. It has this conserved region of DNA, which is an RNA gene that is conserved in all species and is essentially the same, but it's also flanked by these 2 variable regions, which will help us identify on the species level who is in the soil. So after our polymerase chain reaction, we take that product and we send it off to get sequencing so that we can identify the species. For the results of our nutrient analysis, here on the y-axis we have the concentration of phosphorus in the soils. On the x-axis we have the tree status, infected and uninfected trees. As you can see, the infected trees had significantly less phosphorus in their soils. Same thing for potassium here. Concentration of potassium on the y-axis, tree status down here on the x. Infected soils slightly decreased again, but still significant. And then for our nitrate levels, infected trees also had decreased concentrations of nitrates in their soils. You may have noticed that our results differ somewhat from the Ryan Schilling study that he did with Dr. Duran in 2015 with ponderosa pine, where our concentrations of all our nutrients were decreased in infected trees in their soils. In Ryan's study, phosphorus and nitrates were actually increased in infected soils. And we think this is because, like I said, the effects are varying from tree species to tree species. And in ponderosa pine, dwarf mistletoe can heavily defoliate the tree, and oftentimes it contributes a whole lot of organic matter directly under the tree, which would increase these two nutrients here. So, because the dwarf mistletoe is still in nutrients, we should see a shift in the community structure of those communities. Unfortunately, we're still awaiting our results from our DNA analysis, so we have a big cliffhanger here. But we did get some preliminary findings, kind of confirming what we expected. Our soils of uninfected trees are dominated by ascomycota and basidiomycota. And those are the two divisions of fungi that create ectomycorrhizal relationships. So that was expected, but it shows that we're on the right track. And unfortunately, when we sent off our DNA to get sequence, we only got one of our treatments back with good sequence, was the uninfected. We didn't get any good sequence on our infected trees, so we couldn't really compare the two infected soils versus uninfected. But we do expect those results to come in, and when we do, we expect the infected soils will have a switch in dominance from being dominated by ascomycota to infected soils actually being dominated to basidiomycota. Why do we expect this shift? Like I said, they're competing for these nutrients to get to the tree, because the tree has a higher need for those nutrients because of being parasitized. So some of the species are going to be more efficient at that than others and will become more prevalent. Here we have some evidence of that. This Rasula and Soleus species here are very prevalent in the uninfected tree soils, but in the soils of infected trees they're virtually absent. This probably may reflect the fact that Rasula and the Soleus species are less efficient at gathering and transporting nutrients to the tree. Inversely, this Piloderma and Cortinarius species are reversed and in uninfected soils pretty much absent, and then in the soils of the infected trees very prevalent. This could mean that these two species are more efficient at getting those nutrients. Also, it could mean that because they're getting less carbon from the tree, because it's being parasitized by the dwarf mistletoe, the tree is giving less sugar to the fungal community on the roots. And it's been shown that some species can kind of supplement their carbon needs by eating organic matter in the soil, or possibly decoupling from the ectomycorrhizal relationship completely and moving out in the soil and getting their carbon from organic carbon from the tree litter and stuff that's in the soil. Here's some evidence of that. Researchers took all the species that they found in the soil of infected and uninfected, this is in ponderosa pine, and they tested to see how many carbon substrates they could use, and the rate at which they can use them. And they did find that infected soils of infected trees were able to use more carbon substrates or organic carbon litter instead of off the tree roots. And they were able to use those carbon substrates at a higher rate. This evidence is backed up by further evidence where they did an enzyme activity analysis. All these enzymes here are used to break down organic carbon like cellulase and lignin that is found in organic plant matter. The infected trees, as you can see, had much higher uses of these enzymes, which is evidence that possibly some of these species have capabilities of supplementing their own carbon needs. Instead of relying on the tree so much, they can go into the soil and take carbon from the soil. Here's some evidence of the community switching at the fungal root interface directly attached to the roots. You can see here that a low parasitized tree has a pretty diverse group of fungi. There's relative abundance of all these species of fungi here. But upon infection, the community converges into these 3 geopora species. These species are probably generalists that are better at gathering nutrients for the tree and possibly need less carbon from the tree. So they're able to stay on the root more. Interestingly, here's comparing herbivory and competition. Low herbivory, low competition. Community structure looks much the same, very diverse. Upon high herbivory and competition, that same convergence is seen into these 3 geopora generalists. So why does the community change? Like I said, the dwarf mistletoe is parasitizing the tree, and the tree is requiring more work done by the fungus to acquire those nutrients in the soils and bring them back to the tree. And that's why we see these changes here. Another big effect that this might have is if these species can move out and use organic carbon, it's going to increase CO2 output into the atmosphere. Researchers have shown almost a 2-fold increase of trees of infected trees. Their soils put out twice as much CO2 as uninfected trees do. Future directions for this research. Obviously, I want to finish that genomic analysis to identify all the species that are present in the soils. I'd also like to do more broad of a spatial and temporal species analysis, where we look at species present at the roots and all the way through the transect, instead of just at the 4-foot mark. And also seasonally, to see if these changes can be seen from summer to spring or fall to winter or stuff like that. I'd also like to do a carbon substrate analysis on the soils associated with Pinion Pine. That other one that we saw was in lodgepole pine. I'm just curious to see what the fungi are doing with the Pinion Pine. And also along this same note, but somewhat unrelated, the ectomycorrhizal fungi are hard to cultivate, and actually a lot of the gourmet species that we love to eat, like boletes and chanterelles, are ectomycorrhizal fungi. And if we can induce them to eat saprophytically, where they use organic carbon instead of needing that host, that may get us closer to being able to cultivate those gourmet mushrooms that are hard to cultivate right now and virtually impossible. Somewhat unrelated, but kind of interesting. Here's my references. Can I answer any questions?
So I have a question.
Yes.
If you could go back to the slide with the Ken Culling study, the first one, that one.
Mm-hmm.
So what tree, what association was
This
that
is
with?
lodgepole pine.
So this was lodgepole, and then the very next one was also lodgepole?
Also lodgepole.
Oh, okay. So,
And the effects of dwarf mistletoe on that lodgepole pine are pretty severe, just like the ponderosa. They defoliate and slow the growth and eventually kill the tree. Yes?
So do you have to wait until the dwarf mistletoe sort of erupts from the surface to be able to identify if the tree's infected or not infected? And if so, is there any benefit to knowing kind of earlier in that life cycle of the mistletoe what its effects on the fungal community is at its roots?
It probably would be beneficial to be able to identify infections sooner. But because their life cycle is so spread out and it takes 2 years before you even see that swelling, I don't know how we can make that beneficial to be able to identify it and then get rid of it. There are some controls to get rid of the infection, but not 100%. Short of clear cutting the stand, some chemical controls, but I don't think any of those controls are 100% at controlling. Anyone else? Thank you very much.